Hello students, today we will be continuing with the second second session of the chapter Principles of Inheritance and Variation. So today we will be studying the laws. We will be studying today only the first law, that is a monohybrid cross, and we will be studying about the chromosomal disorders. Gregor Johann Mendel played a vital role in discovering the laws of heredity. He carried out several hybridization experiments with true breeding pea lines which had sharply contrasting characters. One such experiment was the crossing of a tall and a dwarf pea plant. Since the cross was between the plants that differed in a single trait, so that is in height, so in this case Mendel called it as the monohybrid cross. So later, Mendel called the tall and the dwarf plants in this experiment the parental or the P generation. After the cross between the tall and the dwarf plants, so Mendel collected the seeds and he planted them to produce the first hybrid called the F1 generation, or it is called as filial of plants. He found that all the plants in the F1 generation were always tall. See, like one of the parents, and the dwarf has been disappeared. So Mendel conducted similar experiments with other pairs of traits and he made some observations. So every single time he found that the F1 hybrid, it resembled only one of the parents, while the other parents trait was missing. To explore this further, what he did, he self-pollinated the F1 hybrids to produce a second hybrid generation of plants. So it is known as the F2 generation. So what he found is, this time he found that the progeny included both the tall and dwarf plants in a definite ratio. That is 3/4 of the plants in F2 hybrid, they were tall, while 1/4 was the dwarf. So he also noticed that the clones did not show blending or mixing. That is, none of them were in between height. So once again, to confirm this observation, Mendel he conducted some experiments with other traits. So his results were confirmed. He found both the parent and all the traits, they were expressed in the F2 generation, that is in 3:1 ratio. So moreover, the contrasting trait, it did not show any blending in either F1 or F2 stages. So from his experiments, so Mendel inferred that something was been stable, which was passed from the parents to the progeny over some successive generations. So this was the reason the dwarf was disappeared, and in F1 hybridization it was reappeared. So Mendel called this one as the factors, so which are known as genes. So genes are represented by the letters per our convenience. So the trait expressed in the F1 hybrid is denoted by a capital letter, while the other trait, it is denoted by the small letter. So as all plants, it obtained the cross between tall and dwarf plants. They were tall in the F1 hybrid. So the tall trait is denoted by the capital letter T and the dwarf by the small letter t. So here the capital T and the small t, they are the alleles of each other. So that is, they are pair of genes representing two alternative forms of the same character. Namely, that is the height of the plant. So therefore, the pairs of the alleles for the height in plants would be capital T and capital T. So capital T and small t small t here, capital T and capital T and small t small t. These are the genotypes of the plant and the descriptive times tall and the dwarf. So these are the phenotypes. The alleles in the genotype capital T and small t are similar or homozygous, while the alleles in the genotype capital T and small t are dissimilar or heterozygous. So moreover, Mendel also called the character that expresses itself in the F1 hybrid are dominant, and the one that failed to express itself is called the recessive. So therefore, through his experiments with tall and short pea plants, Mendel found the character of tallness was more potent over the character of dwarf in F1 hybrid. And the both tallness and dwarfness appeared in 3:1 ratio in the F2 hybrid type. So Mendel called this law of dominance. So the law states that in a monohybrid pure line cross between parents with contrasting traits, only one form of the trait, that is the dominant form, appears in F1 generation, and both forms appear in a ratio of 3:1 in the F2 generation. Also from his experiments, Mendel he observed that the recessive parent trait was expressed without any blending in the F2 hybrid. After verifying the results of the crossings, it was found that the segregation of the alleles is a random process and the chances of a gamete containing either allele is 50%. So this is known as the law of segregation, and it states that the alleles do not show any blending, but instead they segregate from each other during the gamete formation into different gametes. Both the characters are governed by the alleles and are recovered in the F2 generation. So through one of the character is not displayed in the F1 generation. So this is a diagram which is very useful in predicting the outcome of the particular cross or breeding experiment, that is the Punnett square. So this was devised by the British genetist Reginald C. Punnett. So here Punnett square, it is a graphical representation that helps to calculate the probability or the possible genotypes of the progeny. So in a genetic cross. Now let's see how Mendel has used this Punnett square in his experiment. So in the F1 generation, Mendel crossed homozygous dominant capital T and parent with homozygous recessive small t and small t. The gametes produced by the parents are written on the two sides of the square, and the capital T small t progeny produced by the fertilization of the gametes is written with the square. Though the genotype of the F1 hybrid is capital T small t, its phenotypic character is tall. Plants of the genotype capital T small t produced by the F1 hybrid are then self-pollinated to produce gametes of the genotype capital T and small t in equal proportion. So from the square, now you can see that all the pollen grains of the genotype capital T have a 50% chance of pollinating the eggs of the genotype capital T as well as small t during the fertilization. So similarly, pollen grains of the genotype small t have 50% chance of pollinating eggs of genotype capital T as well as small t during fertilization. So thus random fertilization, it results in the zygotes out of genotypes capital T and capital T capital T and small t, and small t and small t. So from the Punnett square, you can also see that 1/4 of the random fertilization they have led to capital T and capital T, half has led to capital T and small t, and finally 1/4 has led to the small t and small t. So therefore, now genotypic ratio will be is 1:2:1. So and 3/4 of the plants are tall and 1/4 of the dwarf. The phenotypic ratio will be 3:1. So the ratio 1:2:1 or 1/4 or 1/2 of the capital T is to capital T to small t is to small t is condensable to a binomial expression of the form ax plus by plus the whole square. So the expression has the gametes bearing the genes capital T or small t in equal frequency of 1/2. So this expression can be expanded as shown. Now we can calculate the genotypic ratios using mathematical probability. So however, by just looking at the phenotype of the plant, it is not possible to predict its genotype. Now we cannot say that whether the tall plant of F1 or F2 hybrid is homozygous dominant. So that is capital T and capital T, or is heterozygous dominant, that is capital T and small t. Therefore, a test cross or a cross between individuals of the F1 generation with a homozygous recessive parent is carried out to determine whether the genotype of the individual is homozygous dominant or heterozygous dominant. And thus, through this monohybrid cross experiments, Mendel proposed the laws of dominance and segregation. Now, these are the chromosomal disorders. So, see, now can you observe this child? Face is swollen, eyes are stunting and bulging, and he has a small mouth with a protruding trunk. So these are the few symptoms of the chromosomal disorder that is known as Down's syndrome. So normal human means that we are having 46 chromosomes arranged in 23 pairs. So however, even if there is a slight variation from this pattern, it will cause abnormalities or some disorders. So chromosomal disorders are caused either due to changes in chromosomal number or due to changes in chromosomal structure. So changes in chromosomal number occur due to non-disjunction of chromosomes. So which is a failure of chromatids to disjoin during cell division, leading to the aneuploidy or to euploidy. So aneuploidy is a condition where one or more chromosomes are either gained or lost. Aneuploidy it is of two types, trisomy and monosomy. A normal diploid individual has 2n number of chromosomes. However, when an extra chromosome is added, the condition is known as trisomy. So that is 2n plus 1 condition. So on the other hand, when the chromosome is lost or when a chromosome of any pair is absent, then it is called as monosomy. That is 2n minus 1 condition. Now, euploidy, it is a polyploidy condition where more than two haploid sets of chromosomes are formed due to the failure of cytokinesis after the telophase stage. The number of chromosomal sets added euploidy can be triploidy or a 3n condition, tetraploidy or the 4n condition, pentaploidy of 5n condition. Same way it continues. Now let us see what are some chromosomal disorders, how it takes changes in number. So Down syndrome or the trisomy 21, it is caused due to the presence of the extra copy of the 21st chromosome. Instead of 2, you are having 3 there. So it was Dr. Langdon Down. He's a British doctor. Okay. He was the person who first described this disorder. Hence the name came Down's syndrome. So the symptoms are the swollen face, bulging and slanting eyes, a small mouth, and a protruding and furrowed tongue. The tongue will always be outside. Some other common symptoms of this disorder are short stature, height will be small, and small round head, a broad palm with a single crease, and the retarded physical, psychomotor, and mental development. Another disorder caused by changes in chromosome number is the Klinefelter syndrome. So it's a condition where males have an additional X chromosome, which results in the karyotype of 47, that is XXY. So Klinefelter syndrome is also known as XXY syndrome, and it is named after Dr. Harry Klinefelter. So he was the first person to describe this disorder. So it is one of the common most sex chromosome disorders in males and shows symptoms such as poor beard growth, appearance will be like female, narrow shoulders, gynecomastia, or it's called as breast development, and underdeveloped testes. Moreover, such males are sterile. And another syndrome due to changes in chromosomal number is a Turner's syndrome, where one of the X chromosome is absent in females. Chromosomes in these females will be 45 with XO. The syndrome is named as a Henry Hubert Turner. He was an endocrinologist and he was the first person to describe this syndrome. So females with this syndrome, they show symptoms like they'll be having webbed neck, constriction of the aorta, they will not have good breast development, and underdeveloped ovaries, and short stature. The height will be small. So moreover, such females are usually sterile and have health problems like diabetes, hearing and vision problems, and also hypothyroidism. So just like changes in chromosomal number, any change in the chromosomal structure or arrangement of genes can also lead to chromosomal disorders. So changes in chromosomal structures can take several forms. Some of these changes include a portion of a chromosome getting deleted or duplicated, means multiplying, or copy of that, or they might be transferred to another chromosome, like getting exchanged. So in that way. So while some of these structural changes in chromosomes are inherited, others takes place accidentally, like when reproductive cells have been formed or during early fetal development. Jacobson syndrome and Friedreich syndrome are some of the disorders caused due to changes in chromosomal structure. So therefore chromosomes hold the genetic keys to all the functions in our body. Any change in number of the structure or the number of chromosome, it will lead to the chromosomal disorder. So these are the important questions. This chapter actually contains lot of questions, children. Each part of this chapter is very important. So first question, state the law of dominance. Second, explain law of segregation. That is, along with the Punnett square, you have to explain that. You have to state the law. And third one, what is Down syndrome and write the symptoms. What is Klinefelter syndrome? Write the symptoms. What is Turner syndrome and write the symptoms. Thank you.